Welcome to G Fun Facts Online. Today, we are going to dismantle the most sophisticated manufacturing plant on Earth. For billions of years, the living cell has been the ultimate factory. Inside these microscopic structures, DNA acts as the blueprint, RNA as the messenger, and ribosomes as the assembly lines, churning out the proteins that structure life, drive metabolism, and defend against disease. It is an elegant, resilient, and self-replicating system. But for a bioengineer, the living cell is also a fortress of frustration. It is a black box protected by a cell wall and governed by complex regulatory networks. Its primary directive is survival, not production. When a scientist tries to force a bacterium like E. coli to produce a therapeutic antibody or a novel enzyme, the cell often fights back. It might degrade the foreign protein, form insoluble aggregates known as inclusion bodies, or simply die because the product is toxic to its own metabolism. The cell's resources are split between keeping itself alive, such as replicating its DNA and building membranes, and making the product the scientist wants. It is akin to trying to build a Ferrari engine inside a busy, crowded restaurant. The environment is simply not designed for the task. This is where cell-free protein synthesis, or CFPS, enters the picture. CFPS represents a paradigm shift in biotechnology, moving from farming cells to engineering biological reactions. Ideally, it is the concept of ripping the engine out of the car and placing it on a workbench. By lysing the cell and extracting only the machinery required for transcription and translation, specifically the ribosomes, enzymes, and cofactors, scientists create an open, accessible reaction environment. In this test tube, or perhaps on a paper strip or in a microfluidic chip, the constraints of life are removed. There is no cell wall to cross. There is no DNA replication to support. There is only the pure, direct conversion of genetic information into functional protein. This technology, once a niche tool for deciphering the genetic code, has matured into a powerhouse capable of producing grams of protein per liter, synthesizing complex therapeutics on demand in remote locations, and even serving as the foundation for artificial life. To understand how this works, we must look at the central dogma of molecular biology. DNA flows to RNA, which flows to protein. In a living system, or in vivo, this flow is tightly regulated by the cell's need to maintain homeostasis. In a cell-free system, or in vitro, the dogma is stripped to its mechanical essentials. A typical CFPS reaction requires 4 categories of components. First is the machinery. This is usually a crude extract, or lysate, derived from cells. It contains the ribosomes, which act as protein printers, the aminoacyl tRNA synthetases, which act as loaders, and various translation factors. Second is the blueprint. This is the DNA template, or mRNA, that codes for the desired protein. Third are the building blocks. This is a supply of the 20 canonical amino acids, nucleotides like ATP and GTP, and tRNAs. Fourth is the fuel, specifically an energy regeneration system. Protein synthesis is energetically expensive, so the system needs a way to regenerate ATP from ADP. Common energy sources include phosphoenolpyruvate, creatine phosphate, or purely metabolic intermediates like glucose. When these components are mixed at the right temperature and pH, the magic happens. The RNA polymerase transcribes the DNA into mRNA, and the ribosomes in the lysate latch onto that mRNA to begin stitching amino acids together. Within minutes, the desired protein accumulates. Because the reaction is open, the researcher can interact with it in real time, adding labeled amino acids or folding chaperones, a level of control impossible in a living cell. The story of CFPS is actually the story of molecular biology itself. It began in the late 19th century when Edward Buchner ground up yeast cells and showed that the resulting juice could ferment sugar into alcohol, proving that biological chemistry didn't require a whole living cell. However, true cell-free protein synthesis had to wait until 1961. That year, at the National Institutes of Health, Marshall Nierenberg and Heinrich Mattei created a cell-free extract from E. coli. They added a synthetic RNA composed entirely of uracil. The system churned out a protein chain made entirely of phenylalanine. With that simple experiment, they cracked the first letter of the genetic code, determining that UUU codes for phenylalanine. CFPS was the tool that allowed humanity to read the language of life. After the genetic code was solved, CFPS was largely relegated to the back shelf for decades. The yields were abysmal, often just micrograms per milliliter, and the reactions would die out after 20 minutes, due to the breakdown of energy sources and the accumulation of toxic waste. For a long time, putting plasmids into living E. coli was the undisputed king of protein production. The renaissance of CFPS began in the late 1990s and early 2000s, driven by pioneers like Alexander Spirin and James Schwartz. Spirin developed the continuous exchange cell-free system, which used a dialysis membrane to constantly feed in fresh nutrients and remove waste, extending reaction times from minutes to days. Schwartz and his team at Stanford meticulously mapped the metabolic pathways active in the E. coli lysate. They realized that by genetically modifying the source strain and changing the energy source to leverage oxidative phosphorylation, they could stabilize the reaction. Suddenly, yields jumped from micrograms to milligrams, and then to grams per liter. The toy had become a factory. Today, we have a toolbox of different CFPS systems. The choice of the chassis, or the organism from which the extract is derived, dictates what kind of proteins can be made. The most popular is the E. coli system. It is the workhorse, cost-effective, robust, and capable of high productivity. However, it cannot perform mammalian post-translational modifications like glycosylation, and the extract can contain endotoxins. It is best for simple proteins and enzymes. Then there is the wheat germ extract, derived from the embryos of wheat seeds. This eukaryotic champion has a unique advantage. Wheat germ contains very little endogenous mRNA, meaning the background noise is low. It is capable of synthesizing very large complex proteins that E. coli chokes on. For mammalian mimics, we use rabbit reticulocyte lysate, made from the immature red blood cells of anemic rabbits. These cells are specialized to produce massive amounts of hemoglobin, so their translation machinery is hyper-efficient and mimics the human folding environment well, though it is expensive. Finally, there is the pure system. This stands for protein synthesis, using recombinant elements. It is the ultimate reductionist approach. Instead of using a crude soup of cell lysate, the pure system is built by purifying every single component individually, all 20 tRNA synthetases, ribosomes and factors, and mixing them together. It is essentially Lego biology. You know exactly what is in the tube, making it perfect for synthetic biology and building artificial cells. So why go through the trouble of brewing proteins in a test tube when bacteria grow for free? The answer lies in the limitations of life itself. The first major advantage is the tolerance of toxicity. If you ask a bacterium to produce a pore-forming toxin to kill cancer cells, that toxin will likely punch holes in the bacterium's membrane, killing the host before it produces any significant amount. In CFPS, there is no living membrane to kill. You can synthesize potent cytotoxins, antimicrobials, or viral components without issue. The second advantage is direct access. In a cell, the reaction environment is sealed. In CFPS, it is open. Need to add a cofactor? Pipette it in. Need to change the redox potential to help disulfide bonds form? Add a buffer. This openness allows for rapid optimization. This leads to the third advantage, speed. To test 1,000 variants of an enzyme in cells, you have to clone DNA, transform bacteria, grow colonies, induce expression and purify, which takes days. In CFPS, you can PCR the DNA variants, mix with lysate in a plate, and incubate. The total time is less than 8 hours. This acceleration of the design-build-test cycle is revolutionizing synthetic biology. Furthermore, CFPS allows for an expanded genetic code. Life uses 20 amino acids, but chemists have created hundreds more. Putting these unnatural amino acids into proteins allows for new chemistries, like proteins that stick to metals or last longer in the blood. Living cells often reject these intruders, but CFPS systems readily accept them. These theoretical advantages are now translating into tangible, world-changing applications. Perhaps the most visually striking application is in point-of-care diagnostics, the Just Add Water revolution. This method involves freeze-drying the cell-free machinery onto a small slip of paper. These papers are stable at room temperature for months. When a drop of patient sample, such as saliva or blood, is added, the water rehydrates the machinery. If the target pathogen's RNA, like Zika or COVID-19, is present, it triggers a reaction that produces a color change. This brings molecular-grade diagnostics to rural villages without the need for electricity or cold chain. Another critical application is on-demand therapeutics. Supply chains are fragile. Whether it is a battlefield or a future colony on Mars, transporting refrigerated medicines is a logistical nightmare. CFPS offers a solution. Transport the lightweight, stable freeze-dried machinery and the digital DNA instructions. An astronaut on Mars with a bacterial infection could download the genetic sequence for a novel antimicrobial peptide, synthesize the DNA, inject it into a CFPS kit, and have a dose of medicine within hours. DARPA is already funding briefcase-sized devices capable of this. In the realm of synthetic biology, researchers are using CFPS to build artificial cells. They encapsulate CFPS reactions inside lipid vesicles. These vesicles can express cytoskeletal proteins to change shape and genetic circuits to compute information, moving us towards self-replicating artificial cells that can clean up oil spills without the risk of evolving. CFPS also solves the problem of difficult-to-express proteins. Membrane proteins are the targets of 60% of modern drugs, but are notoriously hard to study because they clump up when removed from a cell. In a cell-free reaction, researchers can add nanodiscs, which are patches of lipid bilayer wrapped in a protein belt. As the membrane protein is synthesized, it inserts directly into the nanodisk, remaining soluble and functional for drug screening. The field is currently moving at breakneck speed. Recent years, specifically 2024 and 2025, have seen the convergence of CFPS with artificial intelligence. A CFPS reaction has dozens of variables, and optimizing this experimentally is slow. Recently, researchers have used machine learning algorithms to predict the optimal cocktail for specific proteins, achieving massive yield increases that human intuition would have missed. Industrial scaling is also becoming a reality. Companies like Sutro Biopharma have built facilities with 100-liter cell-free reactors to produce clinical-grade antibodies, moving CFPS from discovery to manufacturing. Despite the hype, challenges remain. Cost is still higher than traditional fermentation, though the use of glucose as an energy source is mitigating this. Post-translational modifications in cell-free systems are finicky compared to mammalian cell cultures. And scaling up to huge volumes presents engineering challenges regarding oxygen and nutrient gradients. The future of protein synthesis will likely be hybrid. Commodity chemicals will stay in fermentation tanks, but high-value, complex or personalized products will move to cell-free. Imagine a future hospital pharmacy with a bioprinter instead of shelves of drugs. A doctor could prescribe a personalized cancer vaccine. The sequence is sent to the printer, and a cartridge containing cell-free extract creates the vaccine in hours. This is the promise of cell-free protein synthesis. It is the democratization of biology. By removing the cell, we remove the barrier between digital design and biological reality. We are no longer just herders of bacteria. We are the architects of molecular machines. The cell wall has fallen, and the possibilities are limitless.